Hello students, I'm Sonali Kelkar from City International School, and today we are going to start the chapter How Do Organisms Reproduce Part 2. Let's have a recap first. What is reproduction? Now, reproduction is defined as the production of a new generation of individuals of the same species. Same species is very important because the species can interbreed among themselves, right? So same species is very important. The production of new generation of individuals of the same species that are physically independent of their parents. Now why reproduction is such an important life process? It is not important for the survival of organism, but it is important for the survival of the species on the earth. Okay. And what is the other importance of reproduction? Continuity of life on the earth. Life won't able to continue on the earth if the reproduction is not there. You can able to see the different varieties of plants, different types of animal, different species of animal on the earth that is possible only due to the reproduction. Then the reproducing organism create new individuals which look exactly similar to their parents. There is a difference between similarity and identical. Similar means they are structurally similar, but they are not identical. Identical means they are chemically also same, but that is not possible. So similar, they are structurally they are similar. The similar copies of the individuals are created by DNA replication. We know that DNA replication is the process which takes place inside the nucleus of each and every cell and responsible for the two types of division, mitotic division and meiosis division. Now, types of reproduction. We have done the type of reproduction, asexual reproduction and sexual reproduction. The basic difference between asexual reproduction, here the two parents are not involved, only one parent is enough for the birth of offsprings. Here in sexual reproduction, two parents are involved producing different gametes. In sexual reproduction, it is not necessary, even not necessary, that two individual parents are responsible for sexual reproduction. That can be seen in hermaphrodite animals, that is earthworm, where the single parent, the single organism have two different organ system for producing sperm and ova. Okay, let's see. Types of reproduction, asexual reproduction, sexual reproduction. Under asexual reproduction, we did fission. Fission, under the fission, we did binary fission where the two new organisms are formed or two new individuals or two new offsprings are formed. Multiple fission where the first nucleus divided into many nuclei and then from each nuclei one new individual has formed. Budding, we did budding in yeast, and then spore formation. Spore formation is also found in fungi, in yeast. Then regeneration. Regeneration is found in different type of animals. In fact, regeneration is found in multiple cellular organism and as well as organism which are multicellular, but their cell formation or the body formation is very simple. So for animals like hydra and planera, their body formation, the cell formation is very simple. So they have a particular type of cell which can de-divide and they can form a one whole organism because of the power of regeneration. But we as a individual, we human beings, we have one organ, that is liver, which have a power of regeneration, in which not a whole human being can form, but one organ can be formed. Lizard was another an example. If the tail of the lizard is broken down, then that particular tail or that particular organ can be regenerated, but not the whole organism regenerated. We are going to study the difference between regeneration and fragmentation in the coming up slides. Fragmentation, in the fragmentation, this takes place in multicellular organisms. Spirogyra is an example where a mature spirogyra can fragment, divide into different parts which can grow as a individual. Vegetative propagation, here we are going to do vegetative propagation today in this session. In sexual reproduction, there is a unisexual organism. That means human beings are there. One organism having a particular sex, that is male or female. That is unisexual organism. In bisexual organism, earthworm and tapeworm, one single organism have both ovary and testes responsible for giving rise to the male and female gametes, right? So we call these type of animals as hermaphrodites. Now coming to vegetative propagation. Vegetative propagation, it is another asexual reproduction in plants. Children, this takes place in plants. Okay, and what do we do in vegetative reproduction? Each and every part of the plant, let's take roots, leaves and stems. Okay, so there can be buds also. So these organs or these part of the plants can be removed from the plant, the parent plant, and if we sow these uh, organs in the soil, then we will get a new plant. So that particular root or the part of the leaf or stem can develop into new plants, right? So this is a vegetative propagation, right? So we are making use of the stems, the leaves, the buds, right? Let's see the different type of vegetative propagation, how we are using, what different uh, parts of the plants we are using for vegetative propagation. Organs of some plants used for vegetative propagations are buds or potatoes. You must have seen small white color buds coming out from potatoes, and especially from the eye part of the potato. There is an eye part of the potato. From that eye part, buds arises, and you can cut that particular part. You can cut this particular part, and then you can sow it in a soil. You will see that after some days, the whole plant has developed. Then rhizome of ginger. Even ginger have buds, and you can cut that particular part, so the whole plant of ginger can be grown. Then bulb of onion. Okay, this is a bulb of onion. We can cut this part, and then if you sow this, you will find that the whole plant of onion has grown. There are certain roots over here which are coming out, and if the bulb has this root, definitely this is going to form a new plant. Then leaf of bryophyllum. This is a bryophyllum leaf. Over the leaf margin, there are certain notches. From there, small adventitious roots comes, and you can see in a mature leaf, you can see that from this notches, new plantlet has arrived. You can remove these plantlets and you can sow them separately. Then you will find that separate different plants are grown because of one single leaf. So these type of reproduction that mostly you will find in plants, that is a vegetative propagation. Now let's see by roots what we can get. Buds develops in thick fleshy roots, okay, which later when put into soil develop into new plants. So this type of propagation by root you can find in roots of sweet potatoes. Then dahlia is a root. Dahlia, this is the flower and this is the root of the flower. So dahlia, then sweet potatoes we have already done, and guava. And by stems you can see here the runners. Okay, runners are grass. You can see the grass is also runner, then mint is also runner, then strawberries are also runner. So grass, strawberry, mint, these are the example of runners. The stems that grow horizontally. Now what are runners? These are the stems. These are the plants where the stems grow horizontally. Now there are artificial vegetative propagation also. The propagation that we saw in the last slides, there can be done naturally. Naturally, a bulb of a onion can be grown somewhere else. Naturally, a potato can be grown somewhere else. But there are certain vegetative propagation, artificial vegetative propagation, which has been used in today's scenario, so as to get a good breed of plant which has a good yield, and plant which is immunity, having good immunity against diseases, and which use less amount of pesticides, insecticides. So these plants and the variety of fruits we can get from these plants, these type of artificial vegetative propagation is mainly used in flower bearing plants or ornamental plants, and even fruits bearing plants. Okay, so let's go to the different type of propagation. Right here, the first one is layering. So in this method, a young branch is bent and pressed into the moist soil. After some time, roots develop from the covered part. This is called layering. So what is done exactly in this? They haven't shown a tree over here, but if you take a plant and the lowest part or the lowest branch of that plant is taken, it is bent down little bit. Okay, it shouldn't be bent so down that it get break, but it should bend down properly. And then what you are going to do, this particular part you are going to cover with moist soil. You should make sure that soil should be moist, right? And after some days, what you will find is, you will find roots coming out from this part of the branch which is covered by the moist soil. And then you can cut this part of the branch, and you can sow the whole part. Okay, you can, this part of the plant, okay, you can cut this part, and then this particular part will grow as a tree. So these techniques are used because if you start growing plant or big huge trees which bear fruits after 5 years, or let's say after 2-3 years, it takes so much of time, right? So these type of techniques are used to reduce the time period. We can get a tree which is quite big before time, and which can bear flowers and fruit much before the expected time, which when it is grown naturally. Okay, so in different type of fruits plants we use such techniques. In apples, in jasmine flowers, in black raspberries, in lemon, in mangoes. Okay, so here we can reduce the time limit, and we can get fruits and flower in these plants much before as expected naturally. Then cutting. In this method, a healthy young branch with few nodes and internodes is cut off, and most of its leaves are removed. Okay, so you remove the leaves of that particular part of the stem which has nodes and internodes. Nodes and internodes are very important. Okay, and you cut that plant, cut that part of the stem, and then you grow it. Okay, now cut end is stuck into some good moist soil. Here also you have to take moist soil. Care is taken that the soil remains moist. The cutting will then develop into roots. The cut part that you have sowed in the soil, you will find that the roots are coming from that cut parts and grow into a new plant. Okay, this method is used to propagate plant like rose, sugarcane and pineapples. Especially rose and sugarcane are in these the rose and sugarcanes plants, this method is used. Now coming to the grafting. So it is a horticulture technique whereby tissues of plants are joined so as to continue their growth together, right? So there, here you can see the stalk. Stalk is that plant which has roots in it. So this is the base plant. Stalk is in fact a base plant, and a cycon is a part of the plant which doesn't have roots or any branches. You just take a part of the stem, cut it obliquely, okay, join them in such a manner that these two pieces are joined properly, okay, and then you have to tie this particular piece, okay. After some time you will find that roots are coming from here and the whole part is grown. Now what is the requirement of all these artificial vegetative technique? Okay, now advantage of vegetative propagation are: maintain genetic stability. The plants produced are genetically similar to the parent plant and have all its characteristics. Then plants raised by vegetative propagation can bear flowers and fruits earlier than those produced by seeds. Okay, so here is something which I have already mentioned. You are cutting short the time. So the time period, you are cutting the time period, and you will get flowers and fruits much before their usual time. It also makes possible the propagation of the plants such as banana, oranges, roses and jasmine, which has less capacity to produce seeds. So if you have heard that banana and oranges, they don't have like seeds producing capacity, much seed producing capacity. So it is better to go for the vegetative propagation in such type of plants. Now tissue culture is another type, another type of technique where new plants are grown by using living tissues. Okay, as you can see in this picture, okay, here we have taken out one tissue, a part of a leaf. Okay, you can take a part of a leaf, a small piece of a stem, a small piece of fruit, a small piece of bud, okay, all different other parts of the plants also can be taken, right? As we know that all these parts of the plant, they are made up of living cells or living tissue. So you have to remove it from that. Okay, the tissue sample is being removed here. They have shown that they have removed this tissue sample from leaf. You can remove it from the carrot also, right? So leaf from there, this tissue sample has been removed. Then it is put into a petri dish. This dish has a lot of nutrients, sucrose, such as sucrose, then hormones, auxin hormone, cytokinin hormone. Then it has vitamins, it has minerals. So it has a lot of extract, which is very good for the growth of this tissue. And once this tissue start growing, it is just growing, this tissue is just dividing into many cells, right? And the group or the cluster of the cells which are undifferentiated, this is known as callus, right? Now this callus, part of the callus is being removed, the callus separated and the single cell cultured. You remove the callus and the single cell will be cultured, right? And then you will get a small plantlet from each callet. After some days you will get small plantlet from each callets, right? And these plantlet, now you can see the plantlet here, right? These plantlet can be further sown in a moist soil. So this technique, from a single small part of a plant, you can get many different plantlets, right? So it is very time saving as well as energy saving, and as well as you are getting a plant of your choice, a plant which has good yield, a plant which has good immunity power. So what are the benefits of vegetative propagation? We have already done the benefits of vegetative propagation. Okay, benefits of vegetative propagation: plant can bear flowers, fruits earlier than those produced from seeds. Growing plants like banana, oranges, rose, jasmine that have lost the capacity to produce seeds, right? So these plants can be grown through vegetative propagation. Then they are genetically similar, and genetical similarity can be maintained in plant through these processes. Then helping growing seedless fruits. It's very important. We can get a seedless fruit through this type of vegetative propagation. These are cheaper and easier methods of growing plants. Now coming to the question answer, what is the importance of copying in reproduction? We know that in reproduction, what is copied? DNA is copied, right? The copying of DNA during reproduction is very important. First of all, DNA copying is important because it ensures that the offspring it looks similar to their parents, right? So when offspring looks similar to their parents, this is due to the copying of DNA, due to the process of copying of DNA. So replication of DNA ensures that each daughter cells formed at the end of the cell divisions receives the equal amount of DNA. That is also there. They receive equal amount of DNA. So the number of chromosomes present in the daughter cell and the number of chromosomes present in the parents are same. Daughter cells, or in a sense, you can say the number of chromosome parents present in offsprings and in parents are same. If DNA won't be copied, then the daughter cell won't receive all necessary genes. The genes are very important for the different traits, very important for the function of the body, for the traits of the body, and for the formation of protein. There may be absence of important proteins which are encoded in DNA. Yes, we know that DNA has a protein encoded system in it. Okay, and because of, if there is absence of DNA, then definitely the important proteins cannot be formed. Then copying of DNA may result in recombination, which will lead to evolution. Of course, DNA goes through many kind of chemical reactions. In DNA, there are many kind of chemical reaction, and that chemical reaction further leads to copying of DNA. So copying of DNA is not a perfect process. There can be certain changes in the chemical reaction, and because of that, there can be certain changes in an organism, and these changes result into variation, and further it leads to evolution. Then transfer of genetic material from one generation to another generation. Okay, so this is also due to the presence of DNA. Then stability of population is maintained. Population stability is maintained through DNA. Now why is variation beneficial to the species but not necessary for individual? See, variation is very important, not important, it is beneficial for the species if the condition goes, if the environmental condition goes worst, or if they change, environmental condition gets changed, right? Then the organism or the individual which has a capacity to make certain changes in their DNA, that can able to survive, or the individuals which has already certain changes, which have already evolved, which have certain changes in their genes, which is helping that organism, that individual, to live or to sustain in the changed environment, will definitely helpful for the continuation of species. So that is why variation is beneficial for the species. Okay, it is beneficial for the species to sustain its life, or to continue its species in adverse condition or in adverse environment. How will an organism is benefited if it is reproduced through spores? See, there are certain points. Reproduction through spores is simple, as well as it is faster mode of reproduction. So many spores are formed during this process of reproduction, and they easily get spread away because they are light weighted, so they can spread by floating air, by water, right, by sticking on animals skin of fur, so they can be spread very easily. So they are very essential for the reproduction. Then spores are covered with thick layer which enables them to survive in unfavorable condition. When the conditions are not favorable, then also spores can survive, and when it becomes favorable, these spores will develop into a new plant. Differentiate between fragmentation and regeneration. So let's see the difference between fragmentation and regeneration. Now fragmentation occurs in multicellular organisms with simple body organization. Here regeneration occurs in fully differentiated multicellular organisms with complex body organization. Let's take an example of human beings. In our body, liver has a power of regeneration. Then in lizard is a reptile which is multicellular and which is fully differentiated organism. It is a vertebrate organism, right? All cells have been differentiated. There are different organ system in this organisms, right? But still a few part of these organisms. Let's take the case of lizard, where the tail has a property of regeneration. If accidentally this tail get cut, or if lizard falls from a wall, it leaves its tail, so that whatever organism is attacking on it will have the tail and just be satisfied with it, and this lizard runs away. So and again you will find that after a few days that tail is regenerated in that lizard. So this is the power of regeneration which occur in fully differentiated multicellular organism. Okay, here I just want to add one thing. In planaria this regeneration is there. So few animals has the power of regeneration, but there are particular cells which are responsible for the regeneration. Now in fragmentation, an organism breaks into pieces and each piece develops into new individual. Here organism breaks after it gets mature, and then it breaks into pieces, and each pieces develops into an individual. In regeneration, organism if breaks into pieces, each piece may or may not develop in new individual. For example, Hydra. Hydra and Planaria. Hydra and Planaria, these are the exceptional. These animals regenerate into complete individual. Hydra and Planaria, these organisms regenerate into complete individual from a single segment of their body part. Whereas human beings and lizard, as we have seen, they have a power of regeneration, but only a particular organ has a power of regeneration. Then fragmentation, third point, no specialized cells are involved in fragmentation. Yes, in regeneration, specialized cells proliferate and forms a mass of cell. The cells from the mass differentiate to form different cell types and tissues. Example of fragmentation is Spirogyra, and regeneration of blood cells in mammals. So this is also an example of regeneration. Thank you student. Practice today's topic. Thank you so much.